Welcome to the lecture series in food microbiology. In this series of lectures, for the last couple of episodes, we have been dealing with foodborne illnesses. This lecture and the previous one, we have been concentrating on one particular organism, a gram-positive spore-forming rod, Clostridium, the various species of Clostridium. We saw with the illnesses which are caused by the presence of either the live organisms, perfringens, or the toxins which are secreted by them in the previous lecture. Here, I am continuing the lecture with the other illnesses which are caused by the other species of Clostridium. This is Clostridium botulinum, a very lethal organism. The toxin secreted by this organism is lethal. This toxin can be used as a bioweapon for biowars. It is that lethal. Even minute amount of this toxin is capable of killing human beings. Botulism. The disease botulism is caused by consumption of food containing the potent toxin botulinum of Clostridium botulinum. This botulin is a neurotoxin and it produces neurological symptoms along with some gastric symptoms. Unless prompt treatment is administered, it is fatal. So like snake venom, which causes paralysis, the neurotoxin, the botulinous toxin or a botulin is also neurotoxic. It causes rapid paralysis of all the muscles, causing rapid death. And we have another case which is called as infant botulism. This happens when infants ingest Clostridium botulinum spores, which germinate, grow, and produce toxins in the GI tract and cause the specific symptoms. If you trace the history of how this Clostridium botulinum is identified, in 1793 in Wilbad, 13 people fell ill and 6 later died after eating blunzen. This blunzen is a sausage made by packing blood and other ingredients into pig's stomach. This sausage had been boiled and then smoked, after which this was considered stable at room temperature for several weeks, meaning it was kept at room temperature for several weeks, and then it was consumed as such without any reheating. Several further incidents or sausage poisoning were recorded in years that followed, usually associated with sausages that contained the animal components other than the muscle tissue. This prompted the local district medical officer, Justinius Kerner, to undertake a study of the disease, which later became known as botulism. Botulus means sausage. Kerner noted that several important features, including the fact that heating was an essential precondition for the development of toxicity of the sausages, and small sausages or those containing the air pockets were less likely to become toxic. So this was actually a very important observation. So initial heating is required for the development of toxicity. So which means that the spores which were dormant could attain germination when heated. So this heating was a prerequisite. Second was air pockets. The presence of air pockets prevented toxicity. And this makes sense because this organism is anaerobic. In 1896, the microorganism responsible was isolated, and this was described by Van Emmergen, the professor of bacteriology at the University of Ghent. And this scientist was actually a student of Robert Koch. Van Emmergen, he established that botulism resulted from the consumption of food containing a heat-labile toxin produced by an obligately anaerobic spore-forming bacillus, and he called this as Bacillus botulinus. And he further demonstrated that the toxin could not be produced in the presence of sufficient salt, it means alkali, that it was resistant to mild chemical reagents, and it was not uniformly active against all animal species, meaning it is specific. So the toxin can act only against specific animals. The cells of Clostridium botulinum strains are gram-positive rods. They occur as single cells or as small chains. Most of them are motile. They are obligate anaerobes and form a single terminal spore. The location of the spore, whether it is terminal, subterminal, central, that is an identification among spore-forming organisms. In this category, Clostridium botulinum can be identified by the presence of a single terminal spore. The cells are sensitive to low pH, which is less than 4.6. They have low water activity and moderately high salt. And in the presence of nitrate, the spores cannot germinate. The spores are highly heat resistant. So to kill the spores, you require a temperature at 115 degrees centigrade. But at moderate heat, they can pasteurize. Cells can be killed by pasteurization. The spores require high heat, 150 centigrade for disintegration. Toxins form during the growth. Strains can either be proteolytic or non-proteolytic. The Clostridium botulinum strains on the type of the toxin which they produce can be subdivided. 6 types are seen: A, B, type C, type D, type E and type F. Among these, type A, type B, type E and type F are associated with human foodborne intoxications. A type are proteolytic, type E are non-proteolytic, but type B and F can either be proteolytic or non-proteolytic. So even among the toxins produced or the types of Clostridium botulinum which can cause human foodborne intoxications, you have a difference, proteolytic and non-proteolytic. At the same time, B and F can either be this. There is no distinguish that this is proteolytic, so this is B, or this is non-proteolytic, this is B. F, B and F can either be either proteolytic or non-proteolytic. The spores of Clostridium botulinum are widely distributed in soil, sewage, mud, in the sediments of marshes, lakes, coastal waters, and in the intestinal contents of animals and fishes. Fruits and the vegetables get contaminated with spores from the soil, fishes from water and sediments, and various other food can be from any of these given sources. Type A and type B spores are more prevalent in soil and sewage and fecal matters of animals. Type E spores are marine environment. So this distinguishes which is the type of food poisoning a person has obtained. Depending upon the food, for example, vegetable, fruits, or if you are eating animals, meat, it can be type A or type B. If it is fish or related products, the most probably it would be type E. The toxins of Clostridium botulinum are neurotoxic, as we said before. In general, the toxins associated with food intoxication in humans, A, B, E and F, they are extremely potent, meaning extremely toxic, extremely poisonous, and a very small amount of toxin is enough to produce the symptoms and cause death. So once consumed, these toxin molecules are absorbed from the upper portion of the intestine through the intestinal wall and spread via blood to the peripheral nerves. So since this is a neurotoxin, the effect has to spread via nerves. So from the upper portion of the GI tract or the intestine, they get absorbed. The intestinal walls will absorb the toxin, and from the intestinal cells, these will spread, the toxins will spread through the blood to the peripheral nerves. The toxins block the signal transfers, irreversibly causing paralysis of all the involuntary muscles. So once the nerves are blocked, see, nerves actually respond to electrical signals. So once the signal transfer is blocked, all the transport gets blocked, causing paralysis. And then the toxins move slowly through the body. The toxins produced by the non-proteolytic strains are not fully activated. So for this toxin to become activated, you require treatment. And this trypsin is an enzyme which is normally seen in the gut, in the digestive tract. So the protoxin which is ingested becomes activated once inside the digestive tract. The toxins are heat labile and can be destroyed in the contaminated food by high and uniform heat, such as 90 degrees centigrade for 15 minutes or boiling for 5 minutes. Radiation at 5 to 7 mRad can also destroy them. Cell growth is necessary for toxin production. At optimum growth temperature, toxins are produced in large amounts. So growth of the cell, the spore has to germinate and then it has to grow and metabolically active to produce the toxin concerned. And at optimum growth temperature, toxins are produced in large amounts. The physiological diversity within the species of Clostridium botulinum is recognized by its division into 4 groups, and the molecular studies based on DNA homology and ribosomal RNA sequences have confirmed this group. So, since there is a physiological diversity within species, this Clostridium botulinum is divided into 4 groups based on DNA homology and the ribosomal rRNA sequences. Group 1. These group 1 strains cannot be distinguished from non-toxigenic species, Clostridium sporogenus. To know the metabolic pathways, Clostridium sporogenus is used in the lab as a safe model because the other species, perfringens or botulinus, are extremely toxic and cannot be used in the lab normally. So Clostridium sporogenus is the model organism among this genus which is used. So the group 1 strains are culturally indistinguishable from the non-toxigenic Clostridium sporogenus. They are strongly proteolytic and will betray their presence in the food by partial disintegration of the product with a rancid or cheesy odor. So these proteolytic organisms, group 1 strains, are proteolytic. So since they are proteolytic, they will break down the proteins which are present in the food. And the breakdown products of the protein gives a specific aroma here. In this case, either rancid odor or a cheesy odor. So if the protein-rich food contains these odors, it is an indicator that it might contain the toxin or the organism. But despite these warning signs, the potency of the toxin is such that the amount ingested on sampling, even a small mouthful which you take as a sample to find the taste of the food, is enough to cause illnesses. So the toxin is so potent that even a taste, a small bit which you take as for tasting is enough to give the foodborne illness. Group 2 strains represent greater potential hazard in chilled food. This group 2 is non-proteolytic with native protein. It can grow and produce toxin to about 3 degrees centigrade. So even at 3 degrees centigrade, this organism can be metabolically active and produce the toxin. And they produce spores with low resistance to heat. And they are also more susceptible to inhibition by salt. 3 types of botulism are recognized: foodborne botulism, infant or infectious botulism, and the wound botulism. Of these, food is involved in the foodborne botulism. As we said, the botulinum toxin is a neurotoxin. So, an enterotoxin like Clostridium perfringens enterotoxin acts locally in the gut. But here, since this is a neurotoxin, this affects the cholinergic nerves of the peripheral nervous system. The typical receptor is the acetylcholine receptor of the peripheral nervous system. Toxin ingested with the food and surviving inactivation is absorbed in the upper part of the small intestine, and it reaches the bloodstream via the lymphatics. It binds to the nerve ending at the nerve-muscle junction. It blocks the release of the acetylcholine responsible for the transmission of stimuli, thus producing a flaccid paralysis. See, at the nerve-muscle junction, you have the acetylcholine receptor, which releases acetylcholine as a signal molecule. Release of this as a signal molecule is responsible for the transmission of stimuli, very important for signal transduction. So when the toxin binds to the receptor and blocks the acetylcholine from getting released, it blocks the whole signal transduction pathway, which subsequently causes paralysis. The initial symptom of botulism occur anything from 8 hours to 8 days, most commonly 12 to 48 hours after consumption of the toxin-containing food. The symptoms include vomiting, constipation, urine retention, double vision, difficulty in swallowing, dry mouth, and difficulty in speaking. The patient remains unconscious until, in fatal cases, shortly before the end, when the progressive weakness results in respiratory or heart failure. Say this is a neurotoxin. So as the toxin spreads throughout the body, slowly all the musculature will be affected, all the nerve endings will be affected. So finally, including the involuntary muscles, everything will become paralyzed, leading to respiratory failure or heart failure. The clinician can do little to mitigate the effect of the toxin already absorbed at the neuromuscular junction or neuromuscular blockade. But sometimes the antagonists, neuromuscular blockade antagonists like 4-aminopyridine, have produced transient improvements. See, once the toxin has spread throughout, this is fatal. Any type of medication is not going to affect, and the clinician or the doctor cannot help you to reduce, in any way to reduce the effect of the toxin. But if the symptoms are caught initially itself, maybe the intensity of the symptoms can be reduced a little bit by using neuromuscular blockade antagonists like 4-aminopyridine. But then this is also only transient. This is not complete reversion. Survival is therefore critically dependent on early diagnosis and treatment, principally by alkaline stomach. We need to wash out the toxin completely, somehow remove the toxin component from the bloodstream completely. Alkaline stomach washing to remove any remaining toxic food. Intravenous administration of specific or polyvalent antitoxins to neutralize the circulating toxin, and mechanical respiratory support where necessary. See, once you know that if it is an early detection that the food has been contaminated with botulinum toxin and you have consumed it, so early diagnosis is very important. Then, so what do? You have to remove all the consumed food from the stomach. So you do a stomach wash. The second is to prevent further spread of the toxin which is already present in the bloodstream. Intravenous administration of specific antitoxin components has to be administered. So this will neutralize the circulating toxin. And then if the respiratory muscles have been affected, you require mechanical support. So early intervention, detection intervention can save the life of the patient. In the lab, the botulinum toxin is in the lab culture, the toxin is produced during logarithmic growth phase as complexes, and it is released into the surrounding medium upon cell lysis. In the smallest of these complexes which are produced in the lab, the M complex, the neurotoxin, is accompanied by a similar sized protein with no apparent biological activity. While the larger L complex, an additional hemagglutinin component is also present. So when you culture Clostridium botulinum in the lab and ask the cells to synthesize, the toxin is synthesized in the logarithmic phase and it gets released into the medium when the cell gets sliced. You have two complexes here, the M complex and the L complex. The M complex, it is a neurotoxin. The release of this neurotoxin or the neurotoxin component is accompanied by a similar sized protein, but the biological activity of this protein is not yet known. The larger complex, which is the L complex, has an additional hemagglutinin component. This neurotoxin is synthesized as a single chain protoxin, which is activated by the proteolytic cleavage to produce a molecule consisting of a light chain, a 50 kilodalton, and a heavy 100 kilodalton chains, which are linked by a disulfide bridge. The neurotoxin is synthesized as a protoxin. For it getting activated, you require a proteolytic cleavage. And on cleavage, it produces a molecule containing two chains, a light chain of 50 kilodalton and a heavy chain of 100 kilodalton. And both these chains are joined together by a disulfide linkage or disulfide bridge. And inside the gut, we have the enzyme trypsin, which is capable of this proteolytic cleavage, thereby converting the consumed protoxin into the active toxin. But if more extensive proteolysis takes place in the gut, This will lead to toxin inactivation. So this picture shows how the inactive toxin, you have the heavy chain here, you have the light chain here. This is the inactive toxin. On proteolytic cleavage, this is the membrane translocase domain of the particular protein concerned. This is the toxin binding domain. So on proteolytic cleavage, this becomes the active toxin. Now you have the entopeptidase which is present. This is the picture of how the heavy chain and the light chain get joined up. This is the inactive toxin. This is the C-terminal end of the protein. This is the heavy chain. This is the light chain. You have the double sulfur here, disulfide linkages here. On proteolysis or on reduction, the S here, see you had two S's here. They became SH group here. This is the toxin binding domain. This is the membrane translocation domain of the protein concerned. And you have the catalytic domain here, which is catalytic domain for the entopeptidase. So this proteolytic cleavage actually results from the inactive toxin to become the active toxin. The endopeptidase then cleaves the components of the docking and the fusing complex of the synaptic vesicle, the vesicle that contains the neurotransmitter acetylcholine. This toxin, the active toxin, now has to get bound to the synaptic vesicle. So what happens is that there is a docking domain for the synaptic vesicle. So you have a docking domain and a fusing domain. This endopeptidase will then cleave these components of the docking domain and fusing complex. So when this is lost in the synaptic vesicle, this is the one which is going to contain the neurotransmitter acetylcholine. So then only if this is active will this acetylcholine is going to get released into the synaptic, resulting in signal transmission. But here, the particular protein attacks and the specific peptide linkage is hydrolyzed. It varies. So what is the protein attacked in the type of synaptic vesicle and what is the specific peptide linkage depends upon the type of toxin, which is like type A, type B, type E or type F. Each has a specific activity. But the ultimate result is that the synaptic vesicle release of acetylcholine is prevented, thereby signal transduction is blocked. Toxin types B, D, F and G each hydrolyze a different peptide bond on the vesicle associated with the membrane protein, also known as synaptobrevin. The membrane protein which is present on the vesicle, we call it as a synaptobrevin. And on this membrane protein, you have different peptide bonds. So different aspects of the peptide bonds are broken or cleaved by types A, B, D, F and G. Types A and type E act on or attack on different linkages on the synaptosome associated protein SNAP25. And C1 degrades both SNAP25 and syntaxin. Even though we call as botulinum toxin type A, B, D, F, G, etc., and all of them get bound to the receptor, the effect or the type of protein cleavage which happens or the peptide cleavage which happens, the type of hydrolysis which happens depends upon which is the type of protein or the toxin we are talking about. So you have the breakage of different types of peptide bonds in synaptobrevin with B, D, F and G. In the case of type A, the synaptome associated protein SNAP25 is the one which is attacked. And if it is type C1, both SNAP25 and syntaxin are degraded. For the types C and D, the genetic information coding for toxin production is associated with the temperate bacteriophage. We have seen that certain bacteriophages are viruses which infect bacterial cells. For the types Clostridium botulinum, which produces toxin type C and toxin type D, if they have to synthesize the toxin, they have to be infected with a specific bacteriophage, a temperate phage. Meaning, this bacteriophage exists in the bacterial cell as a prophage, meaning it is incorporated, the genome is incorporated in the bacterial cell, bacterial genome, and every time the genome replicates, the phage genome also replicates. So this does not cause bacterial cell lysis, this exists. This is a temperate bacteriophage with lysogenic phase. So what happens here is that the information for the production of the toxin is coded in the bacteriophage's genome, the viral genome. It is not present in the bacterial genome here. This is exactly similar to another human pathogen, Corynebacterium diphtheriae. There the diphtherial toxin is synthesized only when the bacteriophage infects Corynebacterium diphtherium. So it is the bacteriophage carrying organism which causes the disease or the pathogenesis. Similar to that, here Clostridium type C and D, the genetic information for toxin production is present in the associated bacteriophage. So here, let's see the effect of the botulinous toxin. So see, this is a synaptic vesicle and you have this is a synaptobrevin complex. You have the various types of botulinous toxins, so type A, B, C, the toxins which are synthesized. This is the syntaxin protein. This is the SNAP25 complex. This is the synaptic vesicle. So the different types of the botulinous toxin interact with the various types of proteins associated with the synaptic vesicle, thereby causing damage to the synaptic vesicle fusion, thereby resulting in the paralysis because the acetylcholine is not getting released. The signal transduction pathway gets stopped or prevented, releasing in the paralysis. Infant botulism differs from classical syndrome in that it results from colonization of the infant's gut with Clostridium botulinum and the production of the toxin in situ. Infant botulism mostly seen in infants aged 2 weeks to 6 months. This occurs especially in the time when non-milk feeds are introduced. And at this stage, the infant's gut microflora is not developed or it is less developed, and the gut microflora cannot compete with the Clostridium botulinum which is growing inside the gut. Honey has been associated with several cases of infant botulism in USA, and in many cases they have found Clostridium botulinum spores in 10% of the samples. So it has been advocated that honey should not be fed to children less than 1 year old. The illness is characterized by neuromuscular symptoms related to those of the classical botulism, and the diagnosis can be confirmed by the isolation of the organism and its toxin from the feces. Clostridium botulinum is isolated. It is usually constitute only a small amount of the total microflora. So in many cases, enrichment of the culture is required. After enrichment in a medium like cooked meat broth at 30 degrees for 7 days, the culture is streaked onto the fresh horse blood or egg yolk. So this is how the colonies look after the incubation period. You have the irregular edge colonies which are projecting out, and they also show lipolytic activity on egg yolk agar. A technique has been described that simplifies this procedure by incorporating an antitoxin into the agar medium so that the surrounding toxin producing colonies are surrounded by the zone of toxin antitoxin precipitate. The most sensitive test is the mouse neutralization test. Actually, what we do is we take the food sample and test for the botulinous toxin. After enrichment, we test for the botulinous toxin in mice. So we have the untreated mice, we have the tryptonized botulinous toxin, we have the heated control. So if the symptoms stay positive in mice, then we have to study by mouse protection test by using appropriate antisera. Mouse should have no symptoms of botulism in the other case if the heated control. So even though this is the ultimate test, normally we don't do this because of danger. We have a large number of detection kits for botulinum like the Biogenentech kit, the PCR kit, or the Biotech Diagnostics which produces the food-proof Clostridium botulinum detection kit. Since this is a lethal, to summarize, since this is a lethal organism, the detection for the presence as well as the presence of toxin in food becomes very important. Hence, it is extremely essential that suspected food samples should be tested before consumption. And also, efficient safety measures have to be taken in delivering the food to different